Welcome to your last lecture presentation for the semester. Today we'll be discussing the female reproductive system. This is going to come from Blossom 27.2. At the very end, I'm going to have just a little thing to say about 28.1. So we'll also be taking a look at fertilization, really not for new information, but more just to clarify some things that have already been said about sperm and the egg. And then there are a couple of slides that I'm going to share also that cover the menstrual and ovarian cycle. And those slides are posted for you just underneath this lecture video link. And there's not that many of those, but we'll do that at the very, very end. So let's get started here with 27.2 female reproductive system. You've already seen this in the lab, and so the anatomy of this should not be difficult. Let's take a look here at a mid-sagittal slice through the abdominal pelvic region. And again, a lot of what we're doing in these last couple lectures is comparing and contrasting male and female. And as you imagine, here the only real significant difference about the abdominal pelvic cavity between a male and a female is that in the female, there is a protruding uterus, right? We still have the peritoneum that is wrapped around all of the abdominal organs. But of course, that peritoneum is going to wrap over the very top of the uterus. And so in the male, of course, that would not be there. And what else do we see here? So we've got the uterus. The non-pregnant uterus is relatively small. During pregnancy, it does become much thicker and larger. The very end of the uterus is called the cervix, and that sort of projects, the neck of the uterus projects down into the vagina. And we can see here too the relationship of the uterus with the rectum and the uterus with the bladder. And we'll see this in a couple of other views, but because the uterus is projecting up into the abdominal pelvic cavity, it does create a couple of sort of dead-end recesses here. And there's one here in between the bladder and the uterus. And there's also sort of a dead end right here between the rectum and the uterus. Of course, we also have, just to give you bearings, this is representing the pubic symphysis. And we recognize that from the pubic symphysis back, right, that there is a series of muscles, the pelvic floor muscles, that urogenital diaphragm that I've described before. And in that line of muscles, you would find the external sphincter for the urethra. And we also see here, of course, the vagina that leads up to the cervix and the uterus. Most of the female reproductive system, as you know, is internal. Really all that is present external collectively is called the vulva. And the vulva, again, is just a name for everything that is visible externally. And this would go from the mons pubis. The mons pubis is the fatty layer sort of right anterior to the pubic symphysis. And then you have the clitoris, and then the labia, both the minor and the major. You'll see labium, labia, minora, and minor and major. Don't get too caught up on the Latin versus English variations on that. If we take a look at the female reproductive system from an anterior view, again, the labia are the external structures, vagina, then we see the cervix, and cervix is smooth muscle and is usually restricted, closed. Once that you get past the cervix, you're up into the uterus. The uterus is held in place by a broad ligament. Recall that we've learned long ago that a ligament is, back in the skeletal system, would be something that connected bone to bone, but ligament is more broadly used in other areas. So a ligament is something that holds a structure to a structure. We're going to see a couple of ligaments here. One, the broad ligament, sort of like a big sheet-like structure that helps to hold the uterus into place. There's also an ovarian ligament, and that is simply a solid structure that connects the uterus to the ovary. And not shown here, but you saw it in lab, and we'll see it in a moment, is the suspensory ligament that sort of holds the fallopian tube up. As you come up into the uterus, there is the opportunity to go left or right. There are two fallopian tubes. Another name for the fallopian tubes are the uterine tubes or the oviducts, and they lead to some finger-like projections at the end called the fimbriae. Now the fimbriae come into very close contact with the ovary, and upon ovulation, the release of an egg, there are some chemical signals that are sent out as the egg is being released, and the fimbriae, the fallopian tube, actually will move to wherever that egg is being released. So the egg is not released from the same place in the ovary month after month. We'll see this in a moment as well, but recall that the follicles are all around the cortex of the ovary. And so it just depends on where the location of that follicle is, and it will erupt from wherever it is around the cortex of the ovary. So in order to catch that egg, the fimbriae and that suspensory ligament allow some movement, as the fimbriae will then hover over the area where the ovulation occurs and grab that egg and pull the egg up into the uterine tube. So everything I just said is here in words, so do make sure that you read through this. Again, the external structure is referred to collectively as the vulva. The mons pubis, that fat pad that is anterior to the pubic bone, that is covered with pubic hair post-puberty, and then the labia majora and the labia minora, which are the protective layers, lips, if you will, right? We know labia means lip. Remember, we had the labrum in the shoulder and in the hip. So the labrum was a lip-like layer of cartilage that extended around, for example, the acetabulum or the glenoid cavity in the shoulder. And likewise, the labia here are sort of lip-like structures that help protect some of the entrance into the female reproductive tract. Also, superior and interior portions of the labia minora come together to encircle the clitoris. This is also referred to as the glans clitoris. Just like in the male, there's the glans penis. The end of the penis, the glans clitoris, is abundant in nerves, much like the male, and also is capable of orgasm or of engorgement of blood. The hymen, you may have heard of the hymen. The hymen is a very thin membrane that partially covers the entrance to the vagina. It had previously been thought that an intact hymen was evidence of virginity, but not, or the other way around, that a broken hymen was evidence that a girl had been abused or had sexual contact. That is not necessarily true. That hymen can break regardless if there's been any sort of penetration. And in the male, we had the bulbourethral / Cowper glands. Those were the pre-ejaculatory mucus-producing, lubricating glands, and females have those as well. They're called the Bartholin glands. More oftentimes, in what I call them in the Green Lab Manual, are the vestibular glands. And vestibular because they're found at the entranceway into the vagina. So here we see openings of the vestibular glands, and again, they are going to be producing mucus and lubricating fluids. You can see here now sort of the arrangement of the clitoris and the prepuce, basically the fold over the clitoris, and you can see that it's continuous with corpus cavernosum, right? Just like we saw in the male that were blood filling for erection, same thing is true here in the erectile tissues for the female. So the vagina, let's take a look at the vagina. It is simply the copulation organ of the female. Again, if you were to ask folks on the street, you know, what's the major female reproductive organ, they would likely say the vagina. But in fact, as we learned in the male presentation, it's really the gonads that are the primary sex organs, but the penis and the vagina are the primary copulation organs. And there are layers to the vagina, no surprise, and the walls are lined by an outer layer that's fibrous. There's a middle layer that's smooth muscle and an inner layer that is mucous membrane. And much like the stomach, there are folds along the vagina called the rugae. And interesting is that the epithelium changes in the vagina. We learned from lab 2 that the vagina has stratified squamous non-keratinized. Remember this? The same thing that's in the oral cavity, down the esophagus, the anus is also lining the vagina. But that is only true post-puberty. So before puberty, the vagina does not have a stratified epithelium. It only has a simple epithelium, one layer thick, which is why any sort of vaginal penetration is so damaging to a female, to a young girl, because the vagina is not yet prepared for that abrasion that comes with penetration. And so it's much more traumatic, much more damaging before puberty. As we've talked about, the vagina is a very acidic environment, and there are bacteria that are found in the vagina that are necessary. They actually are helpful to the vaginal environment and keep that acidity below 4.5. And that 4.5 keeps bad bacteria from growing and also minimizes yeast infections as well. So the ovaries, again, these are the gonads. They are the primary sex organs of the female. There's 2 of them. They are what we'll call homologous. I'll show you a list of homologous structures. In fact, let me just kind of take an aside here for a moment and mention that. Developmentally, male and females are both made of the same tissues. So in the early embryo, recall that there are those 3 layers, right? There's the ectoderm, the mesoderm, the endoderm. And from those 3 layers, all of the tissues of the body are formed. Well, through the early weeks of human development, there is no real difference between male and female. Only if the embryo has an intact Y chromosome will the male structures form. So essentially, all of us are by default. The human embryo, the human fetus, is default female, right? The embryo will form, the baby will form as female unless there's a Y chromosome. And if there's a Y chromosome, then the female structures are restricted and the male structures are allowed to grow. So because the same cells of the body that form the ovary have the potential to become the testis, or the same cells that go on to form the penis form the clitoris, and the same forms that are similar structures that form the scrotum in the male, those flaps of tissues become the labia in the female. So we're going to call these homologous structures. I'll show you a table of these in a few minutes. But we'll see that there are similar, or homo, right? Similar homologous structures between the male and the female. I mentioned earlier that the ovaries are in the pelvic cavity. Remember, the male, the testes start up in the pelvic cavity, but descend down into the scrotum. In the female, they stay up there in the top, and they're supported by a continuation of the peritoneum that connects to the broad ligament. Again, that broad ligament found in women basically helps to suspend and hold all of the female structures in place. Now, inside the ovary, if you were to cut into the ovary, you would find that there is a dense connective tissue covering over the ovary called the tunica albuginea. You saw the same layer. The tunica albuginea was also the layer directly around the testis. And then beneath the tunica albuginea is the cortex. Just like any solid organ, there's a cortical region and a medullary region. So the outer portion is the cortex. And in the cortex, what you will find are a bunch of growing or baby, I'm gonna use the word baby eggs. Okay, these are the oocytes. So all of the eggs that a woman will ever have are found in the cortex of her ovary. And because these cells are formed so early in development and have to hang around protected through the lifetime of the woman, those baby eggs are surrounded by a bunch of support cells called follicles. So we'll talk about the collection. The egg plus these follicular cells is called a follicle. So in the ovary, one finds in the cortex these follicles. And we'll see that these follicles stay dormant through childhood, but then upon puberty and every month thereafter, some of these follicles will begin to develop in the hopes of being released through ovulation. And we'll talk more about that. We've got two cycles to keep track of in this unit. We have the ovarian cycle, which is going to deal with the cycles, obviously, of the ovary. And we're also going to be talking about the menstrual cycle, and that's also called the uterine cycle, and that's dealing with basically the blood flow cycles that occur with the inner layer of the uterus. So let's start with the ovarian cycle. And again, when I get past the Blosson, there will be a couple extra slides that I will share with you that will also deal with helping you see this, I think, a little bit more clearly. So the ovarian cycle is a predictable cycle, and I will be describing a 28-day cycle. Now, the 28-day cycle is considered the average cycle. Some women, however, don't have average cycles. They have shorter cycles, longer cycles, irregular cycles. But I will be, in this semester, all we'll be dealing with is a, quote, normal or average 28-day cycle. And in that 28-day cycle, we're going to see that there's a connection between the oogenesis, the formation of the eggs, as well as folliculogenesis, that is the growth and development of the follicles. So in the male, we described spermatogenesis. Spermatogenesis was the formation of sperm from beginning to end. Recall, it took about 64 days. In the female, we're going to have oogenesis. Now, these two processes together, right, spermatogenesis and oogenesis, are two versions, two types of gametogenesis, right? So gametogenesis is simply the formation of gametes. In the female, we would call this process oogenesis. In the male, we call this process spermatogenesis. This is where, though, there's a huge difference between male and female. Recall that in the male, spermatogenesis does not begin until puberty. When the Leydig cells start to produce testosterone, they start turning on those spermatogonia, and from that point on, sperm are produced through the male's lifetime. In the female, oogenesis is going to begin with some stem cells called oogonia, again, there's a lot of parallels here. Remember, in the male we had spermatogonia, in the female we have oogonia. And here's what's different. The oogonia are actually going to start, they're going to initially start the process of meiosis even before birth. So the oogonia will form what's called primary oocytes in the fetal ovary before birth. These primary oocytes are then frozen, that's my word, but they are frozen in the early stages of meiosis. Okay? They're kind of freeze framed. They just kind of, they get started and then they get stopped. And that process will then begin again after puberty and will continue for some of these early eggs through the woman's reproductive years until she reaches menopause, which is basically when there are no more eggs available to go through this process. The number of these oocytes will decline from 1 to 2 million in an infant. So when a young girl is born, she has 1 to 2 million of these early eggs. By the time she reaches puberty, she'll only have 400,000 or so. And of course, by the time she reaches menopause, she will be down to 0. That reduction in the number of these primary oocytes, these baby eggs, is called atresia. So that's an important word to know, atresia. So the ovarian cycle, a part of it is to ovulate, to release the egg from the ovary. And again, this does not begin until puberty, and this happens around the time that a woman has her first menstrual cycle or her initial menses, and from that point on, a woman will have a cycle again approximately every 28 days. So let me ask you, how many cycles, therefore, does a woman have in a year? How many of these 28-day cycles does a woman have in a calendar year? And I usually hear 12 because there are 12 months, but actually, if you look at it, 28 days, there's actually more like 13 cycles every year. 13 cycles, 13 menstrual cycles per year on average. So let's take a look at what's happening here. Again, this is a little bit different than the male. In the female, there are these diploid cells, 2N, 2 copies of the chromosomes, called oogonium, each one, oogonia, plural. And just like in the male, they are turned on by hormones, and they are going to undergo mitosis. One of those cells will know to replace itself, and the other one will know to continue the cycle. And it will go until right here, where this line is. It just barely gets started, okay? But it gets freeze frozen early on in the stage. Then, and only if, okay, the rest of the process will begin to move when the ovarian cycle begins. That primordial follicle or that baby egg will wake up and start developing. And then the egg doesn't even bother finishing meiosis until and if it is fertilized. Okay, so it's a really interesting difference between males and females here. Now, the other thing that's interesting, recall that in a figure like this for the male, that each of the spermatogonia that undergoes this process creates 4 equally sized, equally viable sperm. Notice here that in this process, only 1 egg is formed. And whereas in the male there were 4 sperm, only 1 egg is formed, and the other 3 are called polar bodies. We're not going to go into the genetics of this, but just know that these are not functional these are not functional cells. These cells just kind of go off and die off through apoptotic events. But only one, that's what I want you to know, only one actual egg is formed from one oogonium. Okay? So the number of eggs is far more limited, isn't it? Like I said up here, by the time a woman goes into puberty, she only has, what was the number, 400,000 or so? About 400,000 eggs at puberty. How many eggs will a woman release in her lifetime? How many eggs will a woman release in her lifetime? Assuming one egg per month, how many eggs does a woman need? Remember, guys are releasing hundreds of millions of sperm every single day. But women, if you are, let's just say from the age of 10 to 50, just to keep it easy. Let's say your first menstrual cycle was at 10 and your last menstrual cycle is at 50. That's 40 years of cycling. And 40 years and about 13 cycles per year. That assumes no pregnancy at all, right? Just constantly ovulating throughout a lifetime. A woman's only going to release about 500 eggs. 500 eggs in her lifetime. Right? That's just vastly different. Vastly different than the numbers for the male. So again, I want you to be just mindful of these differences in the timing and in the numbers. So in the end, right, we become the ovum. The ovum is just the word for the mature egg. Okay, it is the final mature female gamete. And again, it only finishes the process of meiosis if and when the egg is actually fertilized by a sperm and then begins to form the zygote. Remember what I said too, is that the sperm, this is woefully wrong, right, by size. I think I mentioned in the last presentation that the sperm is 85,000 times smaller than the egg. So this is not a reasonable representation. This is what you normally see in drawings, right? You'll see a round egg and you'll see a sperm, but size-wise, this is not at all accurate. The ovum is huge compared to the sperm, and remember why? The egg, and if it's fertilized, right, the zygote is going to travel down the fallopian tube for 5 or 6 days and then is going to implant into the uterus and then has to wait until the early placenta is formed before it's going to receive nutrients from the mother. So the zygote, through the egg, needs to carry the yolk sac and the nutrients to maintain life for 2 to 3 weeks. Okay, so that's why the egg is, the largest human cell is the egg, because if it gets fertilized, it must have the nutrients within it to survive for 2 to 3 weeks before implantation and the placenta is formed. And that's why in this process of oogenesis, the polar bodies become very, very small, and all of the cytoplasm from those polar bodies is donated to the larger one. So there's just one that is formed that's much, much larger. Another interesting thing is that in addition to the nutrients that are necessary to maintain the embryo until implantation and beyond, is that sperm only contribute DNA. Sperm do not contribute any cytoplasm. And if you go back and think about where are the mitochondria in a sperm, and if you look at sperm anatomy, you'll recall that the mitochondria line up along the midpiece, or sometimes called the neck, of the sperm. So upon penetration, the sperm does not donate any mitochondria. It only donates cytoplasm. And it donates the DNA, right? The DNA is in the head of the sperm, but no mitochondria. And so it is possible through genetic testing to look at mitochondria And this may be a little bit more information than you need for the test for sure, but the mitochondria have their own sets of genes. Okay, DNA is found in the mitochondria as well. And so it's possible to train maternal, it's possible to follow maternal bloodlines through mitochondrial DNA testing, because males do not contribute mitochondria to the offspring. So all the mitochondria that you have, everyone in the room, all of your mitochondria came from your mother. Okay, from your mother. A little thing about that here, about this mitochondrial DNA. It's an extremely interesting story, and basically through this we're able to basically program or test back through all Homo sapiens, back to the time of what they call the mitochondrial Eve, right? The first woman, if you will, and that explains. But we can test all this through mitochondrial DNA. Very, very cool stuff. So we've talked about oogenesis, the formation of the egg, and related to that is folliculogenesis, the formation of the follicle. So remember that the egg is this, it's just a cell, and it's sitting in the ovary, needs to be protected. So there are these supporting cells that are around the egg called the follicular cells, and they're just sitting there waiting for puberty. Now, what happens though? Every month post-puberty, mechanisms we really don't understand. Hormones are released, and as a result of that, about 20 or so of these primordial follicles, these little baby eggs, every month wake up, and they begin to develop. It's about a 2-month process, and the primordial follicles, some of them, it says here a few, right? They'll be recruited, about 20 every month, and they will become primary follicles. I'm not going to ask you to stage these or recognize them, but the primary follicles will then become secondary follicles, and as this happens, they get larger, more cells grow around them, and then eventually, of all of these early follicles that begin to develop, one will win the race, and all the other ones will fall off, and the one that wins the race goes on to become the tertiary follicle, or you'll see the word mature follicle as well. This mature follicle is the one that will be released through ovulation. Okay, so 99% of the follicles in the ovary will undergo atresia, will never get a chance to be released, right? 99%, only a small percentage, about 500, right, throughout the lifetime, a small percentage of any of those baby eggs will ever make it out of the ovary through ovulation. So do not memorize these extra details, but here's the idea. So in the early embryo, from development on, there's the egg in the center, right? There's the oocyte, and there's just a little ball of cells around it, and that's a primordial follicle, small, found in the cortex of the ovary. Along comes puberty, and with hormonal signals of puberty, 20 or so of these primordial follicles are randomly chosen. Now, these 20 that are chosen could be in the left ovary or the right ovary. They could be anywhere in either one of the two ovaries. And those 20 or so will start to get a little bit larger. They start getting some more helper cells around them. I'm not naming them because I'm not asking you to know them. So these little helper cells grow, and then those primary follicles will continue to become secondary follicles. All I want you to see is that they get bigger, right? More cell layers gather around them. But as this process occurs, only one of those will become the tertiary. I want you to see something. It kind of happened here really fast. Here's the egg, guys. See the egg? See how big the egg is here? Okay. And this is the follicle in relationship. Down here, guys, this little thing is the egg. And the follicle's huge, right? Huge, 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 huge. So the scale like flipped on us here between secondary and tertiary follicle. All I'm saying is that this is huge, this mature follicle. All the other follicles lost the race, and that's it. They're not going to be released. Then this tertiary follicle, this mature follicle, will be released from the ovary. It'll rupture through the outer membrane of the ovary. And sitting out here when it erupts will be the fimbriae, right? Those fimbriae at the end of the fallopian tubes will catch this egg. And this egg will then be up in the fallopian tube. But here's another interesting story, is that all of these cells that got left behind, so when the egg is released, there's still a lot of these cells that get left behind inside the ovary. Those cells that get left behind inside the ovary become the corpus luteum. Now, corpus means body. Lute, we know, means yellow. This has a yellow tinge to it. In fact, that yellow tinge comes from cholesterol. It comes from lipids. It comes from lipids. And you'll understand why in a minute. But the corpus luteum helps to make progesterone. Progesterone, recall, is a lipid. So the cholesterol that is found in the corpus luteum is necessary for the synthesis of the progesterone that the corpus luteum will release, and I'll describe more in a moment. So if you were to cut into a ovary, a prepubescent ovary, and you simply looked around the cortex, you would find just those little primordial baby eggs sitting around the cortex. None of them have begun to develop. If you looked at an ovary that was post-pubescent, after puberty, and there were some of these follicular layers and follicular cycles going on, you would look around the cortex and you would see that there were some random larger follicles. Those would be primary and secondary, as there would be cycles going on where the mature follicle is being produced. And then if you looked at a post-menopausal ovary, you would see a complete atresia, lack of any evidence of the eggs. But you would see some of these corpus luteal, corpus albicans structures, the leftovers, sort of the evidence that there had been cycles in the past. So don't worry about any of the individual details here. Go ahead and blue that out for you if I'm able. Yeah. So again, this is the 28-day cycle. The primordial follicle becomes a tertiary follicle. That takes about 2 months. This is not a short, this is not, it's not a short cycle. Basically 2 months, kind of similar to the formation of sperm, right? About 64 days. And it all initiates the same, male and female. So in men and in women both, puberty and all of these hormones are initiated when the hypothalamus first releases a hormone called GnRH, gonadotropin releasing hormone. And I'll go ahead and type that out just because it's here. Okay, gonadotropin releasing hormone, GnRH. And you know what this means. This is a protein that ends in IN, right? Trope, recall from the vocab, means to influence. So this is a protein, a hormone, that influences the gonads, right? Gonadotropin releasing hormone. So the GnRH is released from the hypothalamus, and it only has to travel a short distance to the anterior pituitary gland. You know where that is, hanging off from the hypothalamus. And then the pituitary gland will produce gonadotropins, there's two main gonadotropins. There's FHS and LH. So let me also just write those out. FHS is follicle stimulating hormone. And LH is luteinizing hormone. Okay. And I'll put that FSH right here. So we have these two hormones, FSH and LH. And I had briefly mentioned these two hormones as having been involved also in sperm production. And so just remember that men and women both produce all of these hormones. Men produce estrogens and females produce testosterones, androgens. And I'm not going to have you worry about which cells, just like in the male, I'm not going to have you worry about which cells are binding these hormones. So I'm going to put a blue section on this as well. So don't worry about any of the hormones and how they're working and the negative and positive feedback mechanisms that are involved with this. Okay, so that is the physiology of this, and you'll deal with that at another time. What I did mention to you, though, is that after the egg is released, that there is this structure called the corpus luteum, the yellow body, that is left behind. I guess it's better to put it in yellow, right? It's luteum. And it is going to be busy making progesterone. And I will say that because that'll make more sense in a few minutes when we talk about some of the cycles. And the corpus luteum is going to allow for the maintenance of the uterus early in this process. So we'll see how that all works in a few minutes. After about 10 to 13 days or so, 10 to 12 days, the corpus luteum then becomes the corpus albicans. Now you know what these words mean. So the corpus luteum is a yellow body, lutea, and the corpus albicans, albi, white, albino. Once it has used up all that cholesterol, and it's no longer, it's used it all up, all that fat is gone, that cholesterol is gone, and cholesterol kind of has a yellow color to it. That's all gone. Then it degrades to become the corpus albicans, which is a white body which has no function other than basically what I call a scar tissue, evidence of a previous follicular and ovulation event. So this is where I'll be coming back. I'm going to be coming back and talking more about everything I've just said, the hormones and the corpus luteum and the corpus albicans in the addendum slides at the end. Let's now go to the uterine tubes, aka the fallopian tubes or the oviducts, and just let's worry about the anatomy here. So initially, these two tubes, there are the fimbriae at the end, and that's what I want you to know. In the middle of the tube, there's an area called the ampulla, and the ampulla is where fertilization normally occurs. So I'll point that out to you here in a moment. And like most things, there's 3 layers. So let's just take a look at this, and then you can just read through up here. But let me just walk you through this from the figure. So the egg has just been released through ovulation. It could have been released from anywhere around the ovary, and the fimbriae will sort of readjust themselves in order to catch that egg. The egg is now pulled up into the fallopian tube, and the ampulla is right about here, sort of the very, very top, and that's the place where fertilization normally occurs. Recall I told you that the egg is only viable for about 24 hours. It must be fertilized within 24 hours, whereas sperm are viable for 72 plus hours. And so sperm can be present 72 hours before ovulation or could present themselves within 24 hours of ovulation. Now question, is it possible for the egg to not be pulled up into the fimbriae? And the answer is yes, okay? So, not 100% of the time do the fimbriae catch the egg. So it is possible for the egg to sort of wander out here, and it would be out here in the female abdominal pelvic cavity. Okay. Now, is it possible for that egg to get fertilized out here in the abdominal pelvic cavity? And the answer, once again, is yes, because sperm come up through the cervix, come up through the uterus, come up through the uterine tube, and they basically just fall off the cliff. And they're falling out into the abdominal pelvic cavity. Now, the egg could be out here as well. So it is possible for the egg and the sperm to find each other and for fertilization to occur. But out here in the abdominal pelvic cavity, there is no place for the embryo to attach and to establish a placental connection. And therefore, any embryo that does find itself out here will simply die off very, very, very young. And the woman will not know that that there was ever any sort of fertilization moment that occurred. But we just know that that does occur. But that embryo is not going to grow out here because it has no, once the yolk is used up, the nutrients in the yolk, there would not be any more nutritional value or opportunity for the embryo to survive. We see here too that there's a rich blood supply, that there's a uterine artery that comes down and provides lots of blood flow to the uterus, and also there's a vaginal artery that similarly brings blood flow to that region. There are 3 layers of the uterus. I'll go ahead and say this now. You saw this in lab as well. The inside layer is the endometrium. The endometrium. Over here, this is not I'm not going to ask you to know the histology of this, but the inside layer is the endometrium. And then there's a muscle layer, smooth muscle layer, that is myometrium. And then the outside layer around the uterus, all around it, would be the perimetrium. So endo, myo, peri. Metri, remember, means uterus or womb. So we've seen all these terms in our vocab. So the inside of the womb, the muscle layer of the womb, and around the outside, the perimeter of the womb. Little story here about some potential issues with sexually transmitted diseases and things. I will not be covering that. So we can also, I'm going to go and just say right from here on down, right? We certainly don't want bacteria or organisms or pathogens entering through the vagina into the cervix or the uterus. But there are possible diseases where, especially with gonorrhea or chlamydia, where there can be PID, pelvic inflammatory disease, which can be a very serious infection of the reproductive organs and can lead to infertility in women. So these sexually transmitted diseases do have an impact in long-term reproductive health. And there are a number of studies that show that the increase in infertility in the U.S., one of the reasons is undiagnosed issues here with chlamydia and gonorrhea in some women. And so that has caused scarring of the uterine tubes and has increased the rate of infertility in our country. So let's continue our journey. We have the uterus. Again, I've already mentioned the layers. This video goes through those layers as well. And I've already mentioned them, so you can just read through about the perimetrium, the myometrium, and the endometrium. The endometrium is the layer that I want to focus the most on, though. It is the layer that you've heard about. You've heard of a disease called endometriosis. Endometriosis is a disease whereby the endometrium becomes scarred, and as a result, if a zygote is formed and the embryo is coming down the fallopian tube, a woman who has endometriosis has patches, regions of her uterus that are no longer available for implantation. And so the more significant the case of endometriosis is, the more likely a woman will have more difficulty with implantation in pregnancy. It is part of the endometriosis, or sorry, part of the endometrium that sloughs off every month and contributes to the flow of blood. And I'll show that to you in a picture, but we already saw up here a moment ago just how vascular the uterus is. And it doesn't show it here, but these blood vessels penetrate all the way in to that endometrium. And we'll see how that's contributing to the blood flow of menses every month. So now let's talk about the menstrual cycle. Again, I'll have a couple of slides that will show this a little bit differently at the end. But the menstrual cycle, the first menstrual cycle, it depends upon age, but somewhere usually 10 to 12 years of age in young girls. And there are 3 phases of the menstrual cycle, and we'll talk about those 3 phases. In those phases, the uterus is shedding, the bleeding phase, the menstrual phase. It's rebuilding, the proliferative phase, and then there's the implantation phase, where it is thick and ripe and ready for the possibility of implantation. Again, I'll show you other pictures of these in a moment. Again, the timing of all this is variable, but we're talking in this presentation, we'll be dealing with the 28-day cycle. Similar to the 28-day cycle of the ovary, there's a 28-day cycle of the uterus, and they're connected, absolutely connected. So the first 5 days or so of the uterine cycle is called the menses phase. This is the bleeding phase. This is what when the woman is menstruating. Okay, and again, 5 days is the average. And this is the very beginning of the cycle. We'll talk about the hormones that are involved here when I show you the other diagrams. This diagram here, nothing wrong with this diagram. It's a good place to start, but I want you to see this. And I have other diagrams for you in a minute. Well, let's take a look. Here's that 28-day cycle. What I don't like about this particular image is that up here, here's 28 days of the ovarian cycle. Well, why not just make this just as wide, right? So we can see this. So it's 28 days both, right? 28 days of the uterine cycle, 28 days of the ovarian cycle. We talked earlier how the follicles are formed, and then the mature follicle is released, and then the corpus luteum makes progesterone for 10 to 13 days before becoming the corpus albicans. So that is shown here pictorially, but remember that this is about a 2-month cycle. So this is an overlapping cycle whereby the follicle is being formed. And then on day 14 of the cycle, right in the middle, right snap dag in the middle, right? 14 out of 28, right in the middle, that's where ovulation occurs. And then we have the luteal phase, which is the remaining days up to 28. And we kind of start over again. So this is the cycle, right? It looks like a linear thing, but it's a cycle, comes back around. So that's what's happening in the ovary, but what's happening in the uterus? Here's the first 5 days, and that's the days of menses. Okay, first 5 days, bleeding. The endometrium is being lost. Then, from days 5 through 14, it's proliferating, it's regrowing. So the uterus then is regrowing itself back. Look at all those little arteries, right, coming up through here. So they become, the arteries restore, and the uterus, the endometrium becomes thicker again. And then the rest of it is the secretory phase. And if you look, day 14, right about here, here's day 14. And I've said before that it takes about 5 or 6 days for, if the egg is fertilized, it takes 5 or 6 days for the embryo, the early blastula, the embryo, to reach the uterus. So if this is day 14, ovulation, if the egg is fertilized, it's not going to reach the uterus till about day 20 or 21. And notice that by day 20 or 21, The uterus is as thick and ripe and ready as it will ever be. So right about here, day 20 or 21, is where the embryo would implant into the uterine wall. And notice that the corpus luteum up here is busy making progesterone. That progesterone is going to make sure that this uterine wall stays intact and maintains the layer. If there is no pregnancy, if there is no embryo, if there's no implantation, the corpus luteum will die off to the corpus albicans. The hormones, the progesterone will no longer be made, and the endometrium will once again slough off in the menstrual cycle. But if there is an embryo, the embryo itself will begin to produce hormones that will maintain the corpus luteum and maintain the endometrium so that it does not slough off. So I'll come back and I'll show this to you in a little bit larger figure in a few minutes, and we'll walk through all of this again. I'll go ahead and introduce these phases again though. There was the menstrual phase, the menses phase, the first 5 days of bleeding. Then the proliferative phase, that's when the endometrium is re-proliferating, right? It's regrowing. If you hear that the North Koreans are proliferating their arms, right? They are building up their arsenal. So the proliferative phase is simply when the endometrium is re-establishing itself after the bleeding phase. And then the secretory phase is that time when the uterus is waiting for the possibility of having an embryo implant, and that is based upon the corpus luteum that's making all that progesterone, telling the uterus to not slough off and be prepared for implantation. Okay, be prepared for implantation. Again, if no pregnancy occurs during the lifespan of the corpus luteum, it will degrade to the corpus albicans, hormone levels drop off, and the menstrual cycle will once again begin. The breasts are an important part of the female reproductive system. They're considered accessory organs to the female reproductive system. Important for nurturing the youth, obviously. In lab, I did not focus on the breast tissue. We don't have a model of the breast, but basically you can see that the milk production in a non-pregnant breast, there is no milk production, and during pregnancy hormones increase that begin to produce the milk, and the milk is produced similarly to sweat or to saliva or to other glandular secretions. And so the breasts are oftentimes just called modified sweat glands because they really do work very, very similarly in producing milk. And the milk is produced, travels through ducts. All those ducts merge at the nipple and are released for lactation. But we're not going to go into any details on the breast. And also, I'm not going to go over any of the oral contraceptives. You can read about that. And that brings us to the end of the Blossom section here for 28 or 27.2. We'll head over for a moment to 28.1. Okay, I said we were going to go to 28.1, but it'll actually make more sense to go to the PowerPoint notes first. And don't forget at the end of 27.2 to answer these questions, including the labeling activities here in the vocab at the bottom. So let's take a look at these couple of these PowerPoint notes. There aren't many here, guys. Let me just pull this up so you can see this. And let's just walk through a couple extra slides that I really think will be helpful for you. So here we go. I mentioned early in the presentation that there are homologous structures. Again, these are just structures that are deformed from the same embryological tissue. So the ovary and the testes are homologous. The clitoris and the glands of the penis. So the clitoris and the tip of the penis are both the tissue with a lot of nerve endings and sexual arousal. Then there's the scrotum and the labia major. I mentioned that earlier. And then also you've been learning about the bulbourethral glands in the male, but there's also those vestibular glands, also called the Bartholin glands, that are in the female, again, for the purpose of lubrication. Let's just go back and talk about those phases once again, just to make sure that you're clean on this. So first, we're talking about the ovarian cycle. The ovarian cycle. Just think what's happening in the ovary. And you know that in the ovary, that's where you have those baby eggs that become larger follicles and that are released. Okay, so the first 13 days, again, we're talking about a 28-day cycle, the first 13 days of the ovarian cycle is called the follicular phase. And about that, during that follicular phase, there's about 20 of these primordial follicles that become primary follicles. So the baby eggs wake up, and again, they're found in either one of the two ovaries. A woman does not ovulate back and forth. It doesn't necessarily mean that 1 month it's her right ovary, the next month it's her left ovary, and the next month it's back to the right ovary. Basically, whichever one of these 20 primordial follicles wins the race to become the mature follicle is the one that wins, and it doesn't matter if it's on the left or the right ovary. Then on day 14, there's ovulation. So on day 14, that is the release of the mature follicle, usually only one a month. And then the rest of the days are referred to as the luteal phase. Recall that the cells and tissue left behind from the ovulated egg turns into the corpus luteum. The corpus luteum is a yellow body because it has a lot of cholesterol in it, has a lot of fat in it. That cholesterol is a precursor. It's necessary for the formation of the sex hormones that are used. Progesterone is the main hormone produced by the corpus luteum, and it is going to maintain the uterine lining. So basically, the corpus luteum is sending out a signal. So the ovary just released an egg. If that egg gets fertilized by a sperm, then what's going to happen? About 5 or 6 days later, right, there's going to be an embryo arriving at the uterus waiting to become implanted. And the corpus luteum is basically sending out a signal saying, hey, uterus, I just released an egg. There's a chance that it's coming down the fallopian tube. And it will give it about a 10 to 13 day window. And if by the end of the 10 or 13 days, the embryo is not implanted, or there isn't an embryo in the first place, then the corpus luteum will regress to the corpus albicans, and that will begin the menstrual cycle once again. Now, if there is a fertilization, right, if an egg does become fertilized, the embryo itself, right, the zygote and the embryo itself will begin to secrete a hormone called human chorionic gonadotropin hormone. You've probably heard of HCG, right? People are using HCG for different reasons now. But HCG is a naturally occurring hormone that is released from the early embryo. And this is also the hormone that is the basis of early pregnancy tests. So a woman gets an EPT in early pregnancy tests, and this hormone can be found in the urine. And these tests are very, very sensitive to the very low levels of HCG in the urine. And if there is HCG present, then it is a positive for an embryo. But let me just say this. Is it possible to have an early pregnancy test say positive and then do the test again a few days later and it's negative? What happened? It's not that the test was wrong. Initially, there was some HCG. Initially, there was an egg that got fertilized and there was an embryo. But remember, every embryo doesn't have the chance to survive or to attach to the endometrium, to the inside of the uterus. So it's possible, and it happens quite often, that fertilization happened, HCG was produced, but that embryo did not attach properly to the wall of the uterus, and the embryo was lost, and so there was no longer any HCG being produced. So that is the ovarian cycle. And then here are the days of the menstrual cycle, also called the uterine cycle. Again, the first 5 days are the menses, or the bleeding phase. Then the 6 to 14 is the proliferative phase. That's when the endometrium is redeveloping. And then you go to the last phase, which is called the secretory phase. The reason it's called the secretory phase is because the corpus luteum is secreting progesterone. Okay, the corpus luteum is secreting progesterone, and that progesterone again is increasing the vascularization and also, making that uterine lining ready and prepared for the implantation. Again, if there is no zygote, no embryo, then the corpus luteum will degenerate to the corpus albicans, and the menstrual cycle will begin once again. What does this look like? I'm going to go jump down to the last slide. The last slide simply puts these two phases, these two cycles side by side, and shows you simply sort of the overlapping. So this is just a nice table to help with the timing, the phases of both the ovarian and the uterine cycle. And let's just take a look at this image. It's similar to what was in the Blossom, but what I like about it is that across the bottom we have our 28-day cycle, and across the top we have our 28-day cycle as well. So it's on the same time scale. And remember that on the top, this is our ovarian cycle. We're forming our follicles. This is the follicular phase. Then on day 14, we have ovulation. And then we have the luteal phase for 10 to 12, 13 more days after that. Versus in the uterus, the first 5 days or so are the menstrual phase, or the menses phase, then the proliferative phase, and then the secretory phase. Once the secretory phase ends, we once again go into menses. Again, let's think about this. So here's day 14, ovulation. If there is fertilization, it's going to take 5 or 6 more days for the embryo to reach the uterus down the fallopian tube. So 14 plus 6, let's say about day 20. So right about here, day 20 or so, is where the uterus is going to be implanted. We first encountered by the embryo, and notice that this is where the uterus is very ready and ripe. It's got all this blood flow reestablished. It is very, very ready. So if the embryo is able to find a place and implant into the uterus, it now has a few days of insurance, right? And the insurance is coming from the corpus luteum. The progesterone produced from the corpus luteum are saying to the uterus, hey, there's plenty of progesterone here. Progesterone is the hormone that's going to maintain the vascular layers and keep the uterus really ready. And it gives the embryo a few days to implant. Again, now if it doesn't get implanted, then the corpus luteum will regress. But recall also that the embryo itself will start to produce HCG, and HCG will have a similar effect. It will maintain the endometrial layer. So what is controlling all this? Okay, now the last slide from this note set is right here. And here's what I want you to see. And this is very simple. Don't make this hard. Here we are. This is the ovarian cycle, right? Follicular, ovulation, luteal phase. And it's showing two hormones, FSH and LH. This is the follicle stimulating hormone and the luteinizing hormone. All I want you to see is that just before ovulation, both of these hormones spike, right? That's all I want you to see. Right before ovulation, these two hormones spike, LH and FSH. And there are, if women are trying to get pregnant, they will do a couple of things. And one is there's a temperature change that occurs, but there's also some tests for these hormone spikes. And these hormone spikes are right before ovulation. Of course, this is the ideal time for intercourse for pregnancy, right? Right before and at the time of ovulation. Now, what else is going on? Remember I said progesterone's in purple, and progesterone levels are kind of flat, and then right about here they go up. Why are the progesterone levels going up? Remember, that's when the corpus luteum is formed, okay? Remember that after the ovulation event around day 14, the corpus luteum starts to produce massive amounts of progesterone. So this progesterone line is really because there's ovulation at day 14, and then the corpus luteum starts to spit out all that progesterone. That progesterone's job was to tell the uterus to continue to get vascular and ripe and ready. And then if the corpus luteum fades again, then those progesterone levels will go down. So basically, the progesterone levels are very, very similar, if not mimicked, by the uterine lining levels, right? Very, very similar. So I always think the uterine lining levels is basically progesterone levels. Progesterone levels. And then notice that estrogen, the blue hormone here, the estrogen levels are higher before. Okay, they go up again, but the progesterone is the main hormone afterwards, but estrogen levels are much, much higher before ovulation. Now, am I going to focus a lot on the hormones? No, but this is important, and we haven't really focused on hormones this semester much at all. Hormones will become a very important topic that will weave in and out of many conversations within your physiology class, and but I wanted to show this to you. So having done this is really the end of the female reproductive system. Let me close these notes down, and we're going to head over just to a little tiny bit of 28.1 and discuss fertilization. And really all it's going to do is clarify and review a couple of things about egg and sperm. Okay, so here in 28.1, just a couple of quick moments here, just pulling a couple things together from the reproductive system, male and female. So let's just take a look at fertilization. This is truly a fascinating process, and I'm really not going to be telling you anything here that you don't already know. But remember, fertilization is the coming together of egg and sperm. When that happens, the DNA from the sperm is going to penetrate into the egg and will meet up with the DNA of the egg, and that will now restore the genetic information from being haploid. Remember, the egg and sperm are both haploid, each containing only one copy of the chromosomes, and now the zygote, the coming together, the union of the egg and sperm, that now contains two copies of all the chromosomes, and we now say that that egg, or that first cell, the zygote, is diploid. So make sure you know those terms, diploid and haploid. Oops, haploid, get that to go, save it, and the diploid cell is, all the cells in your body are diploid, right, except your egg and sperm, which are haploid. Again, the fertilization usually happens up in the ampulla, the very, very top of the fallopian tube, and then it's a 5 or 6 day journey once the embryo is formed, coming down to implant into the endometrial layers of the uterus. So as we talked about this, but as sperm are released, even upon ejaculation, the sperm are, now by the time they are ejaculated, they are independent swimmers. However, they're not great swimmers. In fact, they don't really become great swimmers until they mix with the fluids from the female. And this process is called capacitation. They become more capable, right? They become more capable of swimming. And basically the fluids in the female reproductive tract strip off some layers around the head of the sperm, make them more able to swim, make them more able to penetrate the egg. So that capacitation process is important. And again, we've talked about this, that the acidity of the vagina is down in the 3 to 4 range. So very, very, very acidic. Sperm don't like that. So many sperm are immediately destroyed by the acidity of the vagina. Others will be stopped at the cervical mucus. Recall that the semen contains prostaglandins. Those prostaglandins are smooth muscle relaxers that help to open up that mucus plug, but still, many sperm will never make it past that. There are also leukocytes that are basically phagocytic throughout the uterus. Throughout the uterus. So again, sperm have a tough time, right? It is quite a journey of sort of like a video game of horrible things, right? Where many things are against the sperm as they make their journey. And then this can take 30 minutes to 2 hours for them to make it all the way up to the egg. Now again, the sperm, there may not be an egg there right off. And so they have to sit around and wait for a little while. And again, they're good for 3 days. Some will say up to 5 days, 3 to 5 days. I usually say 72 hours, but there's evidence that up to 5 days. Okay. And again, the egg is only good for about 24 hours following ovulation. So these are all numbers we've already talked about. Not going to get real fancy about this. I just want you to see this. So here is the egg. Okay, here's the egg. It is surrounded by some protective layers around it. I'm not going to name them. I'm not going to worry about it. And here are sperm. And here, again, the size is way off. But we're seeing that about 6 or 7 sperm are making their way right to the egg. What would happen if more than one sperm penetrated an egg? If more than one sperm penetrated an egg, oftentimes I'll hear someone say, "Oh, there'll be twins." No. That's not how twins are formed. If more than one sperm penetrates the egg, that's called polyspermy. Polyspermy is death, basically, to the embryo. Because if 2 sperm are able to penetrate into the egg at the same time, that means that the early embryo is going to have 3 sets of chromosomes, right? 1 from each set of the sperm and 1 from the egg. And mitosis and cell division has no idea what to do with 3 sets of chromosomes. So that is that's a deadly recipe. So those embryos which are formed through polyspermy are non-viable. So we only want 1 sperm, and yet we know there's so many of them. So here's what happens, and I'm not going to go into the details, but just want you to know that once the first sperm penetrates the outer walls of the egg, there are biochemical changes that happen that trigger a blockage of any additional sperm. So only 1 sperm can penetrate, okay? And then the other ones are blocked. There is a fast block reaction. It basically is a instantaneous change that blocks any other sperm from penetrating into the embryo. And then there's a second phase that actually adds even more protection against this. But you see why, right? This would be a problem. And this happens not only in humans, but this happens in other animals as well. Have you ever thought about it? How in the world do fish reproduce? Right? Mama sprays eggs across the rocks in the bottom of the lake, and then daddy sperm comes by and sprays sperm all over the place. You may never walk out in the lake again, right? You're kind of walking around with sperm and eggs all over the place. You've got dozens or more species of fish. Well, how in the world does a salmon egg find a salmon sperm, right? Or a trout sperm find a salmon or a trout egg? Well, same idea. It only takes 1. So 1 comes together, any other ones are rejected. And also in that, which I didn't mention, is that if it's not the right kind of sperm, right? If a trout sperm finds a salmon egg, then there aren't the right number of chromosomes. And so once again, that becomes a non-viable offspring. So Once the egg is fertilized, we now have the zygote. Remember, the egg doesn't even finish meiosis until and if it is fertilized. So initially upon fertilization, the egg has to still finish meiosis. It will finish meiosis, and then the zygote will flip over and begin doing mitosis. The early zygote will split, right? Go through mitosis, go from 1 cell to 2 cells. Those 2 cells will become 4 cells, and off we go in forming the early embryo. And then we'll just talk about twins really quick. And that is, most of the time, women release a single egg. However, 1% of the time or so, women will release 2 eggs. Okay, they could be from the same ovary, they could be from different ovaries, but 2 eggs are released. Well, if both of those eggs are independently fertilized from 2 different sperm, then 2 separate zygotes will form, and if they both implant, then you are going to end up with dizygotic, right? Dizygotic or fraternal or non-identical twins. Okay, 2 sperm, 2 eggs, basically siblings born at the same time. Not identical. Less common is when the zygote divides on its own early in development, right? You've already had 1 egg and 1 sperm. All the genetic information is there. That early embryo splits. Sometimes early, like at the 2 or 4 cell stage, but more oftentimes the early embryo will split around the 70 to 100 cell stage. That's basically about the time where it's coming down the fallopian tube. And depending upon when this split happens, there might be 1 or there might be 2 placentas. And so I'm not going to go into the details of this, but if it happens earlier, then both embryos would be formed in one placenta. If it happens a little bit, if it happens a bit earlier, then both embryos can develop separately and have their own placenta connections. And I just wanted to mention that fertilization story because I think that's kind of cool, and mention the mono and dizygotic twins. As far as the questions down here, you can certainly entertain 1, 2, 3, 4. Yeah, I think you can answer all these questions down here at the very bottom of 28.1, and that does bring us to the end of this presentation.